So, hello everyone. I will present my work that I have done during my PhD since 2017. I'm in my third year now, and I'm working on UniLectin web database, which is dedicated to carbohydrate binding proteins, and more specifically to the lectins. And this PhD is supervised by Annie Berti from Grenoble at the CERMAV and Frédéric Lisasek from Geneva in PIC team. And it's financed by both laboratory and with an association also to GlycoAlps and the Swiss Institute of Bioinformatics. So first I will go back to what are the lectins, which are proteins with at least one non-catalytic domain that binds to monosaccharide or oligosaccharide reversibly, and it's not an antibody. So there is a distinction between the lectins and the CBM, which CBM are carbohydrate binding domain that are next to an enzyme, and we are not focusing on them. So if we look at specifically the lectins, they can be found in the extracellular matrix, in the vacuole, on the cell surface, and are very important for the interaction between host and the pathogen for the fixation on the cell surface. In the domain of the glyco-bioinformatics, there are many types of tools and database, and lectins and unilectins belongs to the Glycan-binding protein domain compared to CASIM database, which focus on the enzymes, or other database that works more on polysaccharides and glycosaminoglycan, for example. UniLectin also has its place in glycoexpasy network of tools which interacts together thanks to relationship between the glycans. So UniLectin provide information on the lectin and the interacting glycans, which are epitopes and that can be related to other glycans, which are fixated either on glycoproteins or can be free to be recognized. And these glycoproteins have also glycosylation sites, and everything is connected together. So if we take a look at the classification of lectins, which is very important for my database UniLectin, we have 3 levels to classify these lectins with an origin describing it's the historical classification. It's the classification which is used every time lectins has been discovered and crystallized to describe them until now, with the origin either bacterial, plant, animal, fungi, virus, and then we have the class, which has an historical name, which is more or less based on the fold, but not exactly and not always. And finally, the family, which can be sometimes the species and sometimes the gene name. So this classification is not really strict, and it's not really adapted if we take a look at the similarity between the amino acid sequence of the lectin domain. Because in fact, we can have a similarity between multiple kingdoms, or we can have a similarity between multiple folds, but in this case, it's not on the lectin domain. And finally, we can have also similarities between multiple families. So for me, which aims to first classify the lectins on my web database and then to use this classification of the lectins to predict them in all genomes. It's really important that I have a good classification of the lectins. And so the issue was how to improve this classification. Two citations, one from 1988, proposed by Nathan Sharon to use the highly conserved amino acid residues that makes patterns to improve the classification of the lectins and the structurally related families and superfamilies. And in 2014, Zui Fujimoto with Jun Habayashi described that some lectins are distributed in several kingdoms and different lectins share the same protein motif, which is an issue because we want to have lectin groups which share the same type of sequence and fold. So it requires a new classification with rules, which we decided to base on the 3D structure and on the conserved motif. So why base the classification on the 3D structure? It's because in fact there are many types of folds which are more conserved than the sequence. So it's really important and it's really efficient to use first the fold and then if the fold is conserved to try to see if there is a similarity of the sequence between multiple lectin domains. And we have many types of interesting fold in the lectins with beta parallel, for example, the beta propeller, the beta helix, and they are very visual and easy to recognize. So what we have done with Anne is to put every lectin together with their fold, based also on PDBE, SCOP-E and CATH database. And thanks to this, we have a curated fold instead of an origin by domain. And for the second level at the class, instead of using just a name, which doesn't mean always the same thing, we use now 20% of sequence similarity for lectin domains that have the same fold. And finally, the family is defined with now the gene name, and it is based on a similarity of 70%. So for the fold, we had issues such as the beta sandwich lectins, which are all described together, and it makes quite a lot of beta sandwich types. So Anne worked on it to improve and divide the beta sandwich class. And we corrected a lot also thanks to SCOP and CATH when possible. And for all the lectins which are not in SCOP and CATH databases, because in fact they have not all the structures and the PDB. In this case, we choose ourselves as a fold. So we also have at 20%, I also had some difficulties to define properly the class at 20% because in fact PDB structures can contain multiple proteins. And what I mean by multiple proteins is that you can have one protein which is repeated multiple times, but you can also have multiple types of proteins which include, for example, one lectin domains and other types of protein which are not interesting for me. So the PDB files and the PDB structures need to be filtered to keep only the lectin domain. And finally, these lectin domains can also contain histidine tags, which have been used, in fact, to during the protein expression to extract the lectin domain and to fix it on the protein, and it needs to be removed from the sequence. And so finally, if we clean everything, we can have a proper similarity between the lectins. And for some of them, I have also one protein which carry a lectin domain and another domain, which is not a lectin domain, but has similarity to a lectin domain. So in this case, it needs also to be cut by hand. So if we take a look at UniLectin.eu website, I have a front page from which for now are accessible without password UniLectin 3D and ProPlec, and are accessible with the password the lectin prediction and the lectin prediction, and also the different publications which have been describing UniLectin 3D and ProPlec with the green buttons. So first we will take a look at UniLectin 3D database, which provides the curated and classified lectin 3D structure. So if we go on UniLectin 3D, we arrive on the front page, which provides statistics on the number of 3D structures available, the number of distinct lectin sequence, because in fact for a protein with the same lectin domain, we can have multiple 3D structures, and also the number of distinct glycans that interact with these lectins. So a quick search panel also provides a quick search for PDB entries, uniprotease entries, or small keywords. And Sunburst also allows to explore this lectin database based on multiple criteria. So the one at the top provides a browser based on my classification by fold, class, and family. And you can put the mouse over the Sunburst to have numbers of each structure lectin in each panel. And you can click on it to have more details. And below it, there is the Sunburst by origin and species, which use, in fact, the old classification. And finally, the one based on the glycans interacting with the lectins, with in the inner circle the monosaccharide composing the interacting glycan, and in the outer circle the full glycan. If you scroll down, there is then an interacting classification tree. First are only displayed the left column, and by clicking on each box, you can open each fold, and the multiple class will appear. And then you can open each class to have all the families present in each class. And you can also click on the blue search button to have all the structure available for this level of classification. If we take a look at the advanced search panel, which is available at the top of the page, it provides the ability to search specifically for different criteria. For example, you can select a fold and a class which match together, because else it doesn't work, and then search either for the 3D structure or the lectin sequence. So what I mean by lectin sequences is the protein that match the lectin domains and can correspond to multiple 3D structures. So if we take a look for this class at the available 3D structures, we have the two first, which are key lectin team in the human with either one type of glycan or a long chain. And we can have more information by going to the view the 3D structure and information. And so by going in the detail panel, there is provided the 3D structure of this lectin thanks to LightMold viewer. So on the left, it's the whole protein with interacting glycan, and on the right, there is a zoom on all the ligand available in this 3D structure, and you can select each ligand to focus on it. So below there is the information for this 3D structure with the resolution and the associated reference. And for the glycans, I have the IUPAC code and the glycosity code. And related proteins to these lectins are also available based on the UniRef database of UniProt. And external links to RCSB, PDB-E, PDB-Care, Glyc2can, Swissmodel, GlyConnect, and UniProt are available. So you just need to click on the button to go on the dedicated page. So for example, PDB-Care is very interesting because it checks for one 3D structure that the glycan is correctly described. Because for many 3D structures, the glycans are not really clean or well defined. So you need to be careful. And so it's also interesting to go to GlycoConnect and Glycan-binding to have more information on the associated glycans. If we take a look further at LightMold viewer, you have a panel at the top which provides the ability to zoom in to have the view in full screen. And you can also click on the configuration panel to be able to manipulate the viewer to hide, for example, the waters or hide the carbohydrate 3D symbol provided by LightMold to check more specifically the glycan. If you scroll down to have more information, there is a panel that are generated in association with PLIP and Swissmodel, which provide information on the interacting glycan. And the difference with LightMold is that it also computes the interaction between the protein and the glycan. So you have the different type of interaction available, and you can put the mouse over each interacting amino acid residue to see in the 3D view the highlighted bond. Below it is a PDB domain viewer in 2D with all the known information on the sequence. So now if we take a look at the second result, it's another interesting 3D structure. And at the bottom, there is the button to go to Swissmodel. And if we click to go see in Swissmodel, we also get the same information for the interacting glycan. And it also allows, starting from a template in Swissmodel, which corresponds to a known 3D structure, to modelize for other similar proteins. In the case you want to, for example, one predicted lectins, you want to know if it could have an interesting 3D structure. So then if we go back to UniLectin and UniLectin 3D homepage, I have also a Glycan search tool, which is very simplified as the numbers of distinct glycan that the lectins interacts to in my 3D structure is very limited. I have only 2028 distinct glycan. So what I did is I simplified based on the glycoprotein glycan builder that I showed you. I only just kept my monosaccharides present in my glycans, and you can click on each monosaccharide to say I want this and I want this other monosaccharide, and then it proposes all the matching glycans with these monosaccharides. And then you can click on one glycan fixated on the lectins, which are epitopes, to have all the corresponding lectin 3D structure. So if you click on it, it goes back to the advanced search page and put the information in the IOPAC search field, and will search for the matching 3D structure of lectin. And by using the exact motif occurrence button, you can search either for the whole UPAC only, or you can search for IUPAC code that contains these the described IUPAC code. Okay. So now if we take a step back to my PhD, so there is the first step of doing the classification of the 3D structure and putting them in the web database with a proper curated classification. And my objective is to predict the lectins in all genomes. So for this, I need, based on this lectin classification, to generate, in fact, motif for each of my lectin class, which then will be used first to try the prediction on one lectin fold, which is the beta propeller, and then to try the prediction on all the lectins to arrive at the final step of exploring and filtrating all these nice prediction of lectins in all genomes. So now we will take a look at the prediction of the lectins only for the beta propeller fold. So first, the lectins with a beta propeller fold, they are separated in 5 different classes. So I have the tachylectin 2, which is by itself. I have RSL and AAL types of beta propeller, which in fact shared similarity between them based on the sequence. So they are grouped in the same class. I have the tectonin, the PLL, and the PVL. So the question is how to predict the beta propeller in genomes. And for this particular fold that we choose to try first, it's because in fact for each lectin domain there is a repetition from 5, 6 to 7 times of the same pattern, which is repeated. And it's more complicated if you want just, for example, to take one complete repeated sequence and use BLAST to search for similar proteins. It doesn't work very well. So another method, in fact, look at the beta propeller, is to use this repetition inside the sequence to cut, based on the 3D structure, the repetition to extract them and align them together. So I have 48 beta propeller lectin structures, and I have 15 distinct beta propeller lectins for my 5 class. If we take a look at one type of beta propeller, this one has 5 repeats. So in fact, I can extract 5 sequences, and there is a small overlap between the end of the propeller with the first repeat. And so by using these 5 repeats, I can align them together to obtain a conserved motif, which is then used later for the prediction. So now that we have a conserved motif, the other issue is to have proper conserved, proper database of proteins in all available genomes, so that I can use my conserved motif to scan all these proteins in all the species available to identify the lectin. And also, as I have 5 family, I have 5 distinct conserved motifs that I will use to scan the database. So if we take a look at the protein database, there is, for example, RefSec provided by the NCBI, which now has more than 100 million entries and more than 100,000 distinct species. So it makes quite a lot of information, and hopefully there is not lectins in all of them. And as we only use for now the beta propeller and only the one for which we have a 3D structure, it's a really restricted type of set of lectin class, and we will see what is predicted. So all these 100 million of entries need to be analyzed for the identification of the lectins. So there's RefSec, but in fact there is also UniProt. So the two of them are working together and most of the proteins are shared, but still there is some differences. So I also use the protein database provided by UniProt, which cover both Swissmodel curated proteins and TRMBL automatically annotated proteins. And if we take a look at the numbers of entry in UniProt, we have 175 million of entries. So based on these protein databases, I use them to scan for my 5 distinct families. And thanks to this, I predicted 3,800 beta propellers, which need then to be filtered based on the score and the repetition to identify proper beta propeller, which would be interesting for further studies. And if we take a look at the predicted proteins, they are present in both the archaea, the bacteria, and the eukaryota. So I put the predicted propeller lectin in a distinct page on UniLectin.eu website, which is the PropLec module, and it can be used to search for specific beta propellers. But I will show this in further detail during the afternoon demonstration. If you look at the predicted lectins in my 5 domains, so I have most of them in my Prop7 EVF domain, and most, in fact, of the predicted beta propeller are in the bacteria compared to the eukaryote and the archaea. And why? Because in fact, the bacteria are already so represented in the available protein database. So it's normal to predict more bacteria lectins when the database of protein I use has already more bacteria. And so if we take a look at the number of repeats present in each predicted beta propeller lectin, so the reference had 5, 6, or 7 repeats, and in fact one family has only 2 repeats which assemble in trimers to make a 6 repeats beta propeller. So we have repeats at 2, 5, 6, and 7 which are known. And what is interesting is that we also have, for example, results are at 3 or at 4 repeats. So if we take a look further in detail, and if we look at the different classes, there is, for example, the PropLec 5A, which is predicted for a good part at 5 repeats, but we also have lots of prediction at 3 repeats for Proplex 5A, because in fact, in this case, it's generally due to less conservation at the beginning and at the end, and only the 3 repeats at the middle are well predicted. But if we take a look at Proplex 6, which is both in red and in purple, they are predicted at 6 repeats, but also quite a lot at 5 and 4 and 3 repeats. So the question was, okay, so we know that we can have 2 repeats that assemble in trimers. Maybe we can have propellers of 3 repeats that assemble in dimers. Okay. So based on the prediction, we decided to take a look at the 3 repeats results. And by looking at the one with the best scores, we had, for example, one prediction in Cordia, which has the 3 repeats, which compared to the reference are very well conserved, and still has the binding site to the glycan. And so based on it, we choose to try to express this protein and see if we obtain a proper 3D structure of beta propeller. And in fact, next to this, we did try the ETC titration, and we showed that this protein is able to bind to the fucose, and we obtained the crystal and finally the 3D structure, which is composed of lectin domains of 3 repeats that is able to assemble together to form a complete propeller of 6 repeats. So if we take for this family, we have the 3 repeats which assemble in dimers, and the one we known before, the 2 repeats which assemble in 3 mers, and the 6 repeats which makes a whole propeller. Okay. I don't know if I can take a look at the conversation. Yes. So I also see that there is the question, what is the definition of domain in this talk? So what I mean by a domain on a lectin structure is that for me, I am interested in my lectin domain, which will be for example, for a whole protein, the part which is used, the complete fold which is used to recognize the glycan and interact with this glycan. So after the prediction in the beta propeller of lectins, we have the prediction of all the lectins based on my 104 lectin class. So now next to it, the update, I have 108 lectin class. And so the prediction of all lectin is available online, but only accessible on UniLectin by using a password. And so if you want to have an access, you need to ask for Anne and Frederic. So if we take a step back at the classification, I had my class made at 20% of sequence similarity, which are composed for each class of multiple lectins with similarities that can be aligned together to generate conserved motif. So for my 100 class, I have 100 conserved motifs, which are used for the prediction, and all the results are then loaded online on my UniLectin predict module. So I have more than 500,000 predicted lectins, and they are present in almost 40,000 distinct species, which is quite a lot. And if I apply a filtering step based on the percentage of similarities between my reference domain and my predicted domains, and I ask to have at least 25% of the corresponding residue that are conserved. And so if I ask for this, I keep only half of my predicted lectins, which these lectins may be more relevant, even if maybe the other predicted lectins with lower score are also lectins, but we doesn't know, because the more the predicted lectins is further away from the reference, the more chance there is that it has lost the capacity to bind to the glycans. So the 20% of similarity, well, the 20% of identity in one lectin class correspond only on the lectin domain to the similarity between the multiple lectin domains. And so based on it, I have quite a lot of predicted lectins in bacteria, in eukaryota, in the viruses, and also in the archaea, but they are not displayed here. And for the eukaryota, we have prediction in the fungi, in the plants, and in the animals. If we take a look and zoom in only on the bacterial lectins, there is quite a lot of fold available, with most of the prediction for the mixed Lysen domain, quite a large number also for the beta trefoil, the beta barrel, the beta helix. And so it's really interesting because in fact, what we see is that we have fold and predicted lectins present in folds that are not yet described for the bacterial kingdom. So the step further away after this prediction of the lectins in the bacterial and in all kingdoms will be to find interesting ways to analyze it and to make nice publication. So I am already working together with Sten and David McIntyre to analyze the bacterial lectins, and I am also working with colleagues on the fungal lectins. Okay, so that's it for the overview of all my PhD work. So now I will take a look at the question.